Hi everyone. Today I'll be sharing the work we do in collaboration with the NLORM Foundation, where we use Oxford Nanopore sequencing to support the design of personalized antisense oligonucleotide therapies for patients with ultra-rare genetic diseases. Here's a quick overview of what I'll be talking about today. I'll start with a bit of background on ASOs, and then I'll discuss our methods, followed by some results showing how this approach performs across a range of genes and sample types. And first, for some background. Antisense oligonucleotides, or ASOs, are short synthetic strands of DNA or RNA that bind to a target RNA molecule in a sequence-specific way. Once bound, they can either block translation of the mRNA, preventing production of a disease-causing protein, or modulate splicing to either skip or include specific exons depending on the need. The goal of this work is to support ASO design through accurate variant phasing. Short-read sequencing can miss phasing between distant or de novo variants, but long reads can give us full haplotype resolution, essential for determining whether variants are on the same allele. Through our partnership with NLORM, we apply this approach to real patient cases. We generate long-read data and phase variants to help NLORM design personalized ASO therapies for ultra-rare patients. And now on to some methods. Once a sample arrives, we isolate DNA and perform quality control, which is one of the most important steps in the entire process, and I'll come back to that in a moment. From there, we move into library prep, flow cell loading, and sequencing, typically using adaptive sampling. Finally, we analyze the data to phase variants and generate reports to support ASO design. Samples can arrive at our lab in several different forms: saliva, blood, cells, or previously extracted DNA. Each type comes with its own set of challenges. Saliva is the easiest and most accessible option. However, it typically yields shorter fragments, which can limit sequencing performance. Blood tends to produce higher quality, longer DNA, but it can be harder to collect, and DNA stability drops after just a few days if not processed quickly. Cultured cells usually yield high quantities of intact, high molecular weight DNA, but samples are more invasive to obtain. Pre-extracted DNA is something we encounter frequently, but it also introduces the most variability. We sometimes don't know how the DNA was extracted or stored, which can impact overall quality. In every case, we tailor our extraction and cleanup strategies to the sample type. Following extraction, we quantify our DNA on a qubit fluorimeter. This table shows typical DNA concentrations we see across different sample types. Then we assess the purity of the DNA using our nanodrop. 260, 280 values give us a sense of protein and RNA contamination, with ideal values for pure DNA around 1.8. And typical values for each sample are shown here. The 260-230 ratio flags contaminants like salts or residual reagents, and is often where we see issues, especially with saliva, due to mucins and other carryover. Finally, we run each sample on the Agilent femtopulse. This gives us a picture of the fragment size distribution and an indication of how well a sample is likely to perform during sequencing. Ideally, we want most of the fragments to be greater than 20 kb. This femtotrace is from a saliva sample and shows a mix of short and long fragments, with many less than 20 kb. To improve performance, we would do a size selection to help deplete the shorter fragments. In contrast, the traces from both blood and cell extractions look great, with most of the fragments above 20 kb. Without good QC, degraded or impure samples can lead to lower yield, shorter reads, and unreliable phasing, directly affecting how well we can guide ASO design. Here's a summary of the 36 samples that we've sequenced using adaptive sampling for N-lorem so far. To define our target regions for adaptive sampling, we use an in-house script that generates a bed file for the target gene and the 3 listed controls. It's only 0.15% of the genome, but with adaptive sampling, we concentrate sequencing on those regions to boost coverage where we need it. Once our targets are defined, we prepare libraries using the LSK114 ligation kit. Ligation prep is ideal for preserving long fragments, which is critical for phasing and structural variant detection. After library prep, we load the sample onto a Promethean flow cell. We sequence each sample for about 24 hours or until we reach about 15 gigabases of data. At that point, we wash the flow cell and reload it with a second library, allowing us to maximize flow cell usage and sequence 2 samples per flow cell. Next, I'll give a brief overview of our analysis pipeline. Base called BAM files are converted to FASTQ, and reads are then aligned to the HD38 reference genome using Minimap2. CLAIR3 is used to call SNVs and indels, which are then annotated with VEP and prioritized using custom scripts. Sniffles and QtSV are both used to call structural variants, which are later annotated and prioritized using Needler. We also run qDNA-seq for copy number variant detection, followed by annotation with bed tools. For phasing, we use CLAIR3 to phase SNVs and indels, and long phase to generate haplotag BAM files. This is critical for understanding whether variants are on the same or opposite alleles, which directly impacts ASO design. Next, using our custom alignment report, we assess target-specific performance based on yield and quality. This helps us decide whether a run meets our quality thresholds. This plot shows genome-wide depth of coverage by chromosome. And you can clearly see a spike in depth on chromosome 2, which corresponds to our target region for this sample. This slide shows haplotagging for our target regions. You can see clear phasing separation across all targets. Depth across each region is between 55 and 64x, and we see balanced assignment to haplotypes in all cases. Now that I've walked through our methods and analysis pipeline, I'll share a couple of examples of our results, starting with a case where phasing was straightforward and followed by a case that was more difficult. This IGV screenshot shows a clear example of phasing at the target gene. You can see strong haplotype separation with reads clearly divided between the 2 alleles, and long reads spanning multiple heterozygous variants across the region. There were also very few unphased reads. In contrast to the previous example, this sample was much more challenging to phase. 2 pathogenic variants were over 150 kb apart, and there were very few heterozygous SNVs in the middle to help us assign haplotypes. In this case, we were able to phase the region, but it required manual resolution of the haplotypes. This chart shows how different sample types performed in terms of coverage and haplotagging. Blood, genomic DNA, and cell pellets all showed good coverage and phasing with balanced haplotype assignment. Saliva samples had lower coverage and more unassigned reads, consistent with their higher variability and shorter fragment profiles. Overall, these trends underscore how sample type impacts both sequencing quality and phasing success and why we adapt our extraction and cleanup methods accordingly. And here's a summary of sequencing depth across the genes we've targeted. Most genes had greater than 30x depth, with an average of about 54x total per gene, with a balanced distribution between haplotype 1 and haplotype 2. This level of coverage was sufficient for us to successfully phase the variants of interest in all samples. And to wrap up, here are a few key takeaways from our work. Variant phasing can be done reliably with long-read sequencing. ONT offers a clear advantage in regions with low SNV density, thanks to its longer read lengths. This workflow is flexible and works across a range of sample types. And most importantly, good QC at the start makes all the difference when it comes to getting reliable long-read data. Together, these efforts enable confident variant phasing and allele-specific ASO design, directly supporting NLORM's mission to deliver personalized therapies for patients with ultra-rare diseases. Thank you to the NLORM Foundation and everyone at the Miller Lab.